Good morning, everyone. I'm really excited to introduce to you our next speaker. She's an expert in the neural bases of reward learning and value-based decision-making. And really, her work has focused on lots of things that are near and dear to my heart, things that I've worked on. She's really been focusing on how our brain learns to associate cues to rewards, and then how this affects our everyday choices, really. And her work has been published in several top journals, including Neuron and Nature, which really shows how influential her work has been. Notably, our next speaker is not only a rock star scientist, but she's actually really dedicated to diversity and equity and inclusion in science, which is, I think, really special. And she has actually recently started a program called, let me get this right, the Superior Opportunities for Maximizing Access to Neuroscience program at UCLA, otherwise known as SOMA. And this is a program that bridges Hispanic serving institutions to UCLA. And this was started by our next speaker. She got the very first institutional grant of its kind at UCLA, which shows really how dedicated she is to promoting diversity. Not only is she an excellent scientist, but also, dedicated to diversity. She's also a really, really great mentor. And I don't know if she knows this, but at UCLA, several of the graduate students and postdocs will routinely get together to talk behind her back, to say things like how supportive and just really what an excellent mentor she is. And I've been really lucky to have her in our same department, because I benefited from her really, really thoughtful feedback. Just yesterday, I grabbed her over here at the conference to bounce off some ideas on some stuff that I'm working on. And so she's just really an overall great scientist. And so without further ado, I want you all to put your hands together for our next speaker, Dr. Alicia Izquierdo.
I was super curious about my walk-up song, I'll be honest. Like, I love LA or something. That was even better. So thank you so much, Melissa, for that very kind introduction. I just want to thank Mike and Manuela for putting on such a wonderful conference. This is my first time here at this Learning and Memory Conference, and it's been wonderful so far. And I've never been so happy to see poster tubes again. It really is fantastic. So today I'll present work from my lab where we study the neural mechanisms of value learning and value-based decisions. We study decisions involving effort, delay, and risk costs. And we're particularly interested these days in learning under uncertainty, so learning under probabilistic outcomes. And we study this in freely moving, freely behaving rats, which sometimes cooperate and sometimes don't. Okay, so to set the stage, options in the real world are rarely this straightforward, where you have clearly a good choice and clearly a bad choice. So much more often, choices are a little bit in the gray zone and of relative value. So we've been interested in, for example, two good options where one may be qualitatively more preferred than the other. So, if you're feeling optimistic, it's good versus better, but maybe pessimism, bad versus worse. So, for example, to me, these look like two good options. The one here on the left, I can get for very little effort at home. I just press the on button on my coffee maker, and I have my coffee. But this cappuccino on the right, it's my favorite in all of Los Angeles. And I have to say that it's actually kind of risky to get there because I'm on the west side and this is located in Silver Lake on the east side. And so this is just to say that, preferred options usually are associated with steeper costs, right? And so LA traffic is definitely, there's a lot of risk and uncertainty in LA traffic. So we're interested in how choices like these are mechanistically implemented in the brain, but just as important as that, and actually to speaking to a room full of learning experts, we're more interested actually in how those options acquire value over time. So how do we accumulate evidence about that over time, about each of those options, and then be able to make good decisions? So historically, regions of the prefrontal cortex have been studied in this type of learning and decision making. Okay, and there are actually a good number of us working on rodent frontal cortex now, about actually learning about flexible learning and decision making. And it does beg the question, when you're studying frontal cortex in rodent, how does that translate to what we understand about the prefrontal cortex in human and non-human primate? And actually, we could discuss this all day, and we could get into some heated arguments. Actually, this is probably not the forum, but this is a very controversial topic, ongoing debate. But I think it's good to always consider, are we comparing apples to apples? And the short story is that based on the complexity of the prefrontal cortex, we can't make those simple anatomical comparisons and say that they're the same thing. So as you can see on the right-hand side, you see rodent frontal cortex is completely agranular, shown here in yellow, and that is that it lacks a granule cell layer 4. That's the major input from medial dorsal thalamus, and that has historically been, the existence of that layer 4 has historically been the definition of prefrontal cortex in primate. You can also see, though, that if you're looking at the primate, at the macaque prefrontal cortex, the more posterior and medial aspects of non-human primate PFC are agranular and more similar, actually, in terms of connectivity to the rodent. So, perhaps more appropriately, we can rely on those species similarities of genes, connectivity maps, as I mentioned, sort of how everything is wired up, and the similarity of frontal and prefrontal electrophysiology, and of course, highlighting their function, because that's actually what we tend to rely on here, is that functional correspondence or behavior, less on cytoarchitecture or that the layering of the cortex, because that actually is a losing battle. Okay. So I'm showing you here two subregions of rodent frontal cortex, OFC and ACC, orbital frontal cortex and anterior cingulate cortex, which will be the topic of today's seminar. Both highly interconnected with basolateral amygdala. And here are just a few roles that have been proposed for these regions in rat and macaque brain. So there's actually good convergence across species in terms of their functions, so lots of similarities. However, instead of looking for differences and sort of drawing lines in the sand and saying that OFC does this and ACC does that, we've been particularly interested in understanding if there are overlapping functions for rodent frontal cortex. So what, if anything, does rodent frontal cortex generally do? So there may be significant value in directly comparing frontal cortical regions on the same behavior and also seeing how amygdala contributes to that as well. So here's an example of what may be a general function of rodent frontal cortex. So this is actually from a couple of years ago from Alex Stolyarova and Mohsen Rakshan. And the lab at the time was inspired by some really nice evidence from the Kepish lab that suggested that OFC was required for optimal weighting based on decision confidence. So this was a really neat task. Rats made perceptual decisions of compound olfactory cues. So either going left if it's more A, if this odor cue is more A, go right if it's more B. And in a separate step, rats would they were required to report their confidence in that decision by waiting for reward. So that's what the authors called temporal wagering. So interestingly, muscimol inactivation of OFC resulted in a selective impairment in that decision confidence, that temporal wagering, but there was no effect of OFC inactivation on that discrimination accuracy. So we thought this was really cool. We designed a near identical task, but with visual stimuli and touchscreen response. So here the rats had to categorize a visual cue as either more horizontal or more vertical, and in a separate step, wait around for reward. Before performance on this task, Alex infused either ACC or BLA with inhibitory dreads and found an identical pattern, top right, such that ACC inhibition resulted in impaired confidence report. So you can see here that rats don't wait differently for reward based on that signal-to-noise ratio. That's very different than the very top panel where you see that kind of separation in the vehicle control group. But this was interesting because there was a completely intact pattern of discrimination performance. So shown here at the bottom of this figure, of this slide, by probability corrector D prime. So rats could discriminate the stimuli appropriately, but their decision to wait for reward did not track with that discrimination accuracy. So all this to say that OFC may not be special in this ability. So maybe we can conclude actually and sort of rewrite that title from Adam's paper. Frontal cortex, or rodent frontal cortex, is required for optimal weighting based on decision confidence. So in fairness, we also didn't look at OFC or any sort of subregions like any parts of the medial wall, infralimbic or prelimbic. So it could just be that there is more specialization in OFC and ACC and not the rest of frontal cortex. But we thought this was pretty compelling evidence that in a different sensory modality, we find the exact same pattern. So here's another example of perhaps a general function of rodent frontal cortex. So this was co-led by Evan Hart and Garrett Blair. So here we conducted a series of experiments investigating effort-based choices. And typically this involves training animals to work on a progressive ratio schedule for a high-valued sucrose pellet reward. So they love these delicious sucrose pellets. But then in other sessions, we introduce a lower valued reward, such as a ramekin of chow that rats typically get in their home cage. So I see this as kind of, do you want to work for creme brulee or just go for that ham sandwich that you've got packed in your backpack? So at the time, there had been some evidence that OFC, in particular the medial sector of OFC, was important in these kind of effort-related decisions. And Evan and Garrett probed this in ACC, infusing either inhibitory or excitatory dreads in this region, administering CNO before either PR sessions or PRC sessions, finding, actually much to our surprise, that either inhibition or excitation of ACC resulted in a selective decrease in willingness to work in the PRC condition. The PR condition actually wasn't affected, so it did suggest that it was something about the evaluation or the comparison of those options and not so much just the general motivation. So additionally, and not getting too much into the calcium imaging, here, they conducted 1P calcium imaging with miniscopes, and they found that population activity in ACC could discriminate the context of that work. So you can see here the ACC signal is low. This is the bottom panel there. The ACC signal is low in the PRC condition in blue compared to the PR condition in red, and this is population activity leading up to a lever press bout. And I guess the point of this is it wasn't the objective of our experiment to sort of directly compare ACC or OFC, but it does show that ACC does something similar to OFC, and it does provide yet another example of some similar overlapping functions for these regions. So at this point, I was going back to the lab and saying, everything that OFC can do, ACC can do better. But coming back to the schematic, there are a couple of other noteworthy dissociations that you'll find in the literature if you're comparing these regions. So in non-human and human primate, ACC seems to be more involved in action-based learning, whereas OFC is more involved in that kind of stimulus-based learning. And I'm showing you here the supporting papers by Rudebeck and colleagues, and this is all human and non-human primate. So there's definitely evidence of that kind of specialization in primate prefrontal cortex. But there's also another level of sort of these dissociations that you find in rodent lesion and pharmacological inactivation studies. You also find evidence that ACC is more involved in learning of initial contingencies, and OFC really only involved in reversal. So when those contingencies have to be updated or somehow when they change. So we asked if there was a hierarchy of modulation. So for several reasons, we actually think that ACC may be positioned in a way to be sort of at the top of the hierarchy, being closer to action. And so that influenced or that inspired where we put our dreads in this next experiment that I'll tell you about. So the following experiment was led by Dr. Tony Yee in the lab. Tony was an electrophysiologist trained by Steve Cowan at the University of Arizona before coming to my lab. And he was very interested in oscillations in flexible learning and decision making, and actually had a deep interest in Parkinson's disease. And he's now a research scientist at Lundbeck in Denmark, and please feel free to go to see our accepted manuscript, which I'm about to describe. So theta oscillations seem to be critical to value learning and reversal learning specifically, so there had been some evidence already in rats. So Fatahi and colleagues found enhanced synchronization in that theta to low beta, this is 4 to 20 hertz frequency bands in ACC and OFC when rats chose as higher valued reward, and less coherence when they chose a lower valued reward on a T-maze. And in some recent experiments in macaques, Knudsen and Wallace found robust theta alignment during fixation of a reward predicting stimulus, as well as after stimulus exposure. You can see that that was specific to the theta, theta oscillations and not beta or gamma. And further, what I thought was really interesting is that closed loop microstimulation during the trough of theta disrupted reversal learning. So you can see that they did microstimulation at the bottom right. They did microstimulation in different trial epochs and really only found an impairment when they did that closed loop microstimulation at that particular time when the animal was fixated on the reward predicting cue. So this was some of the, I think, the first compelling causal evidence of theta in reversal learning. So collectively, those data show that frontocortical regions may be cooperatively involved in supporting reward learning, and further that this may causally involve theta oscillations. So Tony was really interested in further understanding this modulatory role of theta oscillations in reversal learning, and also combining this with viral techniques. So this is the general timeline down below here for his experiments. Following initial pre-training of his rats to respond to stimuli in the operant chambers, he infused the rats with bilateral inhibitory dreads, or EGFP. So these dreads are on a CAMP-K promoter, although I do realize that there have been some recent reports that the CAMP-K promoter is not as selective as it once was believed to be, but still more selective than the HSYN promoter. Either way, we used a CAMP-K promoter for these, and also a null virus EGFP in ACC. And also in the same surgery, he implanted rats with 16-channel unilateral electrode arrays, targeting both ACC and OFC. So again, the dreads are bilaterally placed in ACC, but the electrodes are targeted both in ACC and OFC. So let me tell you a little bit more about this task. So this is an example implanted rat tracked using deep lab cut by Dr. Andrew Howe. And this is actually faster than real time. So just so you know, the animal's actually a little bit more deliberative than this. So the rat's job is to visually discriminate a rewarded stimulus from an unrewarded one. And there are specific trial events that we typically tend to track. These are trial initiation, choice of the correct stimulus, choice of the incorrect stimulus, and reward collection. So Tony programmed these TTL pulses to precisely align the EFIS with the behavioral events. And this is the experimental design. Down at the bottom here, Tony administered CNO after rats reached criterion in this initial discrimination phase. And this was just to test if ACC inhibition affected that kind of mastery level performance or retention of that initial discrimination. And then he counterbalanced the drug order only in reversal learning. So you can see that half of the animals received vehicle first, and then the other half of the animals received CNO first. So we were really particularly interested in that early reversal learning, which is, you typically tend to see some perseveration, which I'll show you in a minute. So Tony found that ACC inhibition does not affect the mastery of that initial discrimination, but does actually result in an impairment in stimulus-based reversal. So first, let me show you at the top left, you can see that there are no differences in learning in those virus groups. So the HM40I and EGFP learn equivalently, and this is actually pretty important for DREADS experiments because people are seeing some like virus overexpression effects, and so these are longitudinal studies, and so we want to make sure that that there are no pre-existing virus group differences, and there weren't any. And then you can see in the bottom panel that animals, when they're given CNO, when they've already reached criterion on that initial discrimination, there's no problem. There's no difference between the HM40I and the EGFP. So you can see here overall there's an attenuation of reversal learning following CNO, and when you plot by whether animals received CNO or vehicle first, that's when you see this really interesting removal of perseveration after ACC inhibition. So this was actually really interesting to us because the evidence had been, I mean, historically, prefrontal cortex lesions or inactivations actually produced enhanced perseveration and not sort of this random response strategy that animals tend to adopt here. So we think that there are two possibilities for this. Either the rats are newly happy with a 50% reward rate, all right? So great, this is fantastic. I don't have to work so hard, I'll just do whatever. And we don't think that that's the case actually because we have other measures like auxiliary measures to look at like latencies and such that don't suggest that there are differences in motivation. However, the second possibility, which we tend to believe in here, is that they can't remember the previous contingency. They don't have access to a cognitive, what people have called a cognitive map, to inform them of what task they're in, whether they're in discrimination or whether they're in reversal. And so a random response strategy would be very consistent with that idea. Now, if that sounds like what the orbital frontal cortex does, then I would agree. So the Schoenbaum lab and colleagues have a lot of data suggesting that orbital frontal cortex is critical for forming a cognitive map. But here we're showing some evidence that, that ACC may do this too. So let's look at the neural data. These are sample event-triggered theta traces in ACC at the top and OFC here at the bottom. And this is then baseline subtracted theta responses here at the time of choice of correct versus incorrect trials. And what's interesting is that there's no way to distinguish between correct versus incorrect trials from ACC theta in reversal learning following ACC inhibition. You'll notice it's completely preserved. There's a completely preserved theta accuracy signal in OFC following ACC inhibition. And we also find preserved accuracy signals, as you can see in that middle panel, in the EGFP group. So this was all within subject measures of normalized theta power across sessions. And this is where the analysis was actually, the N was sessions. And thanks to a wonderful reviewer comment, here, if we look at individual rat and normalized theta power, again, now here with the dots in the scatter representing animals as a single value per animal, you can see the identical pattern. There's a readout of accuracy in OFC, but no such effect in ACC following inhibition. So maybe there's something that we can learn from the speed of response, and as I mentioned, we looked at latencies as well. So here you can see latencies for each of the trial epochs for discrimination in the top row and reversal in the bottom row, and we saw a significant decrease of incorrect choice latencies in the reversal phase following ACC inhibition. So rats are quicker to make incorrect choices in reversal, which is not a good thing. You want them to be more deliberative. And I don't know if you can make out the beta coefficients there, but we found significant, when we put everything in a mixed effects GLM, we found that significant predictors were OFC theta and also drug exposure for that incorrect choice latency difference. There was no such speeding of incorrect choices in the EGFP animals following CNO compared to vehicle, which is great because what that suggests is that this is not an effective drug, it's an effective ACC inhibition. So I know I have a room full of experts here in terms of theta power in hippocampus. So we knew that theta power in hippocampus is correlated with acceleration and velocity. So we also looked at correlations of theta power and latencies in our task. And we found no significant correlations of either ACC or OFC theta power with those choice latencies, either correct or incorrect. So admittedly, our animals they're not on a maze, they don't have much room to move, so that's sort of a caveat there. We also surprisingly didn't observe any statistically significant coherence differences for correct versus incorrect choices in the reversal phase. So we found ACC OFC theta coherence to be fairly weak in our task, despite seeing these significant differences in theta power during choice accuracy. Okay, so to summarize this part, there does seem to be something special about OFC theta in reversal learning. Indeed, you could read out accuracy in the reversal learning phase via OFC theta, independently of ACC inhibition. On the other hand, ACC inhibition promotes this random response strategy in early stimulus-based reversal learning, suggestive of a similarity with OFC, which has been more associated with flexible learning of stimuli more than actions. Also, ACC inhibition attenuates an ACC theta signal of accuracy and hastens incorrect choices in reversal learning. And this we compare directly with a lack of effect of CNO in our EGFP animals. And as I've suggested, that rats can't really distinguish correct from incorrect trials and respond randomly following ACC inhibition, it does indicate that ACC is also important in forming what some have called a cognitive map like OFC. So for the next part of my talk, I'd like to tell you about some unpublished work by PhD candidate Claudia Aguirre. Claudia's dissertation work is focused on the systems that support flexible learning of stimuli and actions. You can see a preprint here that's currently under review. And I'd like to highlight actually that Claudia has been the first graduate student in my lab that has systematically included both males and females in all of her experiments. So what I showed you before, actually Tony did include males and females, but since it was an electrophysiological study, we were underpowered to look at sex differences. But the other studies that I mentioned, that I described by Alex and Evan, those were all in males. So I think it's been really great. It's been quite eye-opening for me to to have both males and females in our experiments because there are really some pretty cool, I think pretty cool sex differences that I'll share with you today. So coming back to this slide, maybe I've convinced you that there's not as much specialization of function in rodent frontal cortex as once thought. So here at the bottom, I'm suggesting here that ACC can do this too. And Claudia was really interested in comparing the contributions of all three nodes to all three nodes of this network to reversal learning of stimuli and actions. I'll present only a subset of these data that are included in the preprint. The ACC data are actually not quite camera ready, but when I do include it, it's actually the case that we have confirmed placement of our dreads in that case. So I will show you some ACC data to compare with OFC in the next couple of slides. Okay, so to remind you, this is what the stimulus-based task looks like. The difference here in Claudia's experiments is that both deterministic and probabilistic learning is what she assesses in her experiments. So in this case, it really is a choice, not just between good versus bad, but better versus worse. And I also want to point out how difficult it is for animals to reverse here. So I didn't point out when describing Tony's experiments that the rewarded stimulus actually moves left and right. So it's pseudo-randomly could be presented on the left or the right-hand side of the touchscreen. So the rat really does have to track the identity of the stimulus properties. And so they can learn this, but reversals, as I'll show you, are pretty difficult. So she uses another version of this task where rats need to discriminate the better rewarded side, and if you blinked, you missed the slide transition here. This is actually completely unsignaled by any cues. The rat has to just, in this case, go left or go right. And we analyze the same trial epochs, same dependent measures. The only difference is that animals, again, must ignore the properties of the stimuli and make a spatial response. So in a room full of learning experts, you can imagine that this is an order of magnitude quicker for the rats to learn. Rats learn this on hundreds, on the order of hundreds of trials instead of over sessions that each session includes 100 or 200 trials. So it's a lot easier for animals to learn the spatial responses. So these are some of the behavioral measures and the associated constructs that Claudia studies in these tasks. And you can see here that in addition to those omnibus measures of sessions to criterion, we also like to look at trial-by-trial interactions and trial-by-trial indicators of performance, including latencies and omissions. And most of these operational definitions, actually, we borrowed from the 5-choice serial reaction time task. So I've been a fan of that task for decades. And so we're not really inventing anything. These are the same mappings that are used in that kind of attention and response inhibition task. So first, let me tell you about and highlight the sex differences that Claudia is finding, and then we can get into the brain manipulations. So though there are some latency differences in reversal, you'll notice that you don't really see sex differences in the rates of acquisition in the discrimination phase. So you can see that in the top right. Those are the learning curves. You also don't see sex differences in the reversal phase. Both males and females have similar acquisition curves, and what I'm showing you here is actually 2 probability schedules. They're pretty difficult probability schedules. In fact, the most notable difference here is that between discrimination and reversal phases. So stimulus-based reversal is very difficult for the rats. They spend many sessions below chance, and they tend to asymptote just above chance, so like around 60%. So again, coming back to this idea that maybe perseveration is an adaptive strategy, and it actually does signal that the animal has a memory of whatever they experienced previously, so that these are completely intact animals, they don't have dreads. So even though rates of learning don't differ, females adopt different strategies than males. So females tend to stick with their choices of the worst option after they get rewarded more than males, which is it doesn't sound like an adaptive response, but it works for them. And we also find that females tend to be stickier in their choices. So stickier meaning higher perseveration indices than males in the reversal phase, but not in the discrimination phase. So not surprisingly, you don't see a whole lot of perseveration in discrimination in the top row there. So this was really interesting because around that time, there had also been another paper showing actually exactly the same thing. So our data are very consistent with Nicola Grissom's results. Her lab showed that male and female mice reach a similar level of accuracy, but they get there a bit differently. So this group also actually used touchscreen response in mice. And their task is a little different, so they call this a restless two-arm bandit task, where the probabilities change within a session. They get reversals within a session. But essentially, they found the same thing, that female mice tend to explore less, but learn more from exploration. So there's more exploitation in female mice. Males actually tend to get stuck in exploration. Am I doing okay on time, Alyssa? Okay. So the previous slides are actually setting up the potential that we may see some sex differences in Claudia's experiments in OFC and BLA. And indeed, here I'm highlighting that most of the previous work on OFC and BLA in reversal learning in rats was conducted almost exclusively in males. Our lab is guilty as well, you can see there from our 2013 paper. And mostly, these studies have delved into action or spatial-based responding. Mostly pharmacological and lesion experiments, not viral manipulations with DREADS or opto. And also, they've mostly targeted lateral orbital and medial orbital frontal. And this is quite interesting, actually. There's kind of an obsession with this dissociation of medial and lateral orbital frontal, when in fact there are fewer studies looking at VLO, that ventrolateral orbital, that receives just as dense of an input from amygdala. So we wanted to address many of these gaps. So Claudia prepared male and female animals with inhibitory HM40I DREADS on a CAMP-K promoter or EGFP control virus. In these regions, had them recover, and then put animals on deterministic and probabilistic reversal learning of stimuli and actions in counterbalanced order. It was a pretty big project for her doctoral work, and I bet you can guess that there was an order effect. So if animals learned the spatial response first, they found the stimuli-based learning very difficult, whereas if they experienced stimulus discrimination first, they weren't affected and could learn the action-based with no problem. So Claudia found that BLA and OFC are necessary for both deterministic and probabilistic reversal learning. So, given this learning forum, here you have a bunch of learning curves. I realize this is a very busy slide, but the y-axis is the same for everything. So here you see probability correct, which is also probability of the better option. And initial learning is shown there on the left across EGFP and our two HM40I groups. No CNO was given initial learning, but when rats were tested on deterministic reversal 1 and 2, and then probabilistic reversal 3 and 4, you can see that if they were given CNO first, they were really affected. And so you can also see that there was a bigger effect in amygdala on the probabilistic task. So we also found, and you probably can't see the beta coefficients, but we also found that a significant moderator was sex in all of these reversals. So subsequently, we entered sex as a covariate, and we found that females are more adversely affected by OFC and BLA inhibition compared to males. So here again, once we enter sex as a covariate, we find that there are effects of OFC inhibition in female R1 and R3, but not males, and an effect of BLA inhibition in both males and females in R3. So these results, I think, pretty convincingly show that the effects of OFC and BLA inhibition are sex-dependent. We also studied stimulus-based learning, and here you can see that rats learn the initial contingencies, but find reversal very difficult. So to study detection and adjustment of reversal in this slow learning, we compared 3 sessions leading up to reversal and the first 3 sessions of each reversal, and found that OFC, but not BLA inhibition, produced a failure in detecting first deterministic and probabilistic reversal. So this was a very pronounced dissociation between OFC and BLA. And we find similarly that if you look at win-stay-lose switch strategies, essentially the same thing, there's no kind of adjustment during reversal for OFC, following OFC inhibition. Okay, so what about ACC? So we do plan on comparing ACC with striatal subregion contributions, but here's a sneak peek at what ACC is doing in the same stimulus-based reversal task, which does nearly exactly what OFC inhibition does. In fact, probably the only difference is an effect size difference. So coming back to the summary slide, there does appear that there's not a whole lot of specialization in rodent frontal cortex, certainly not as much as you see in the human and non-human primate, and I've just shown you evidence that OFC does action-based reversals too. So not going to read you all, for interest of time, I'm not going to read you all of the summary points because I've pretty much highlighted all of those things, but I will end with that overall we're finding very similar effects for OFC and ACC inhibition in most rat learning and decision-making tasks that we've looked at least in our lab. So the question is, are there any dissociations between OFC and ACC in rat? And I just want to point you back to this slide that I've beat you over the head with. So one reliable finding for ACC is that it signals reward prediction error. So when people have recorded here, they do find RPE signals. So this is the difference between what you get and what you expect. And there's really actually no convincing evidence that OFC neurons signal this at all. In fact, the Schoenbaum lab have recorded in OFC for decades, and rather they signal expected value or some kind of range of expected outcomes, but not individual RPE. So I think we're finding evidence in our ongoing experiments that ACC may keep this more fine-grained reward statistics, particularly in probabilistic reversals, which I just want to point out on the same slide. This R3 is really the first time that animals get a reversal, and it's also the first, a probabilistic reversal. It's the first time that animals are experiencing misleading feedback. So rewarded on the worse option, unrewarded for the better option. So it does suggest that ACC is particularly important in those conditions. As you can see, that is very close to a 50% hugging of the line, even more so than OFC inhibition. So let me just end with some thoughts about theory. So with our collaborator, Alireza Soltani, we have considered how the brain copes with and learns under different forms of uncertainty. And based on the experimental evidence that I provided you today in rodent and primate, we think that at least these 2 cortical regions, and this is where we've looked, that these 2 cortical regions contain some information about expected uncertainty, which Alireza has formalized here at the top right, which I won't even attempt to explain, but which I think is essentially something you can only accumulate over trials or over sessions. So we think that this is a sort of a trial accumulation in ACC and a longer-term session accumulation in OFC. So I think this is the sort of the the function of frontal cortex in rot. So this is similar to what others have proposed about frontal cortex and risk or variance or standard deviation of value. And this kind of information is really important in learning because it's used to gauge when updating is needed, right? To set the rate of adjustment, to let me know when I shouldn't take the 405 and go to get my favorite cappuccino. Okay, with that, I'd like to thank funders, collaborators. I've highlighted the work of these talented individuals in blue, and thanks for all your kind attention.